plant cell. Here sketch of typical plant cell, where you can see nucleus, one of the membrane bounded organelle. Nucleolus is also there. Mitochondrion is another one. Then chloroplast is here. Endoplasmic reticulum. So these are some of the membrane bounded cell organelles. So here you can see in between the nuclear membrane and plasma membrane somewhere structure which lies over here, that we are going to study today in some detail, and that is the Golgi complex. So scientist Golgi in year 1898 discovered this membrane bounded cell organelle he was Italian neurologist. Botanists, they refer this cell organelle as a dictyosome even. How this cell organelle, Golgi complex or dictyosome, look? So that is here in this figure. So you can see here the cisternae, then intercisternal space between these two cisternae, then this is the cisternal membrane, and these are the vesicles. So we will see morphology of Golgi complex, where we will take survey of its shape, size, number and position. So shape of Golgi is quite variable in somatic cells, and even in the same cell there will be variation in the shape of Golgi complex. It also depends on variation of the what functional stage is of that cell. On that also shape depends. In some cases, Golgi complex occurs as dense reticulum of tuberculi, while in others it occurs as hollow spheres united together. Size of Golgi is also variable, may be small or large in different types of cells. Its number per cell is also variable. maybe single Golgi apparatus in single cell, or maybe as many as 100 one can see in a single cell. Position of Golgi complex is relatively fixed for each type of cell. Its position may be in between nucleus and periphery of the cell, or may be found between nucleus and excretory pore. Now, structure of Golgi complex. First electron micrograph of Golgi complex was taken by Dalton and Felix in 1954. Golgi complex consists of three components: flattened sacs, called as cisternae, large vacuoles, and small granules. All these are membranous structures. Or call it as a smooth membranes. Morphologically, Golgi complex lies between endoplasmic reticulum and plasma membrane. Golgi complex consists of about 60% protein and 40% lipid. Here, lipid fraction contains mainly cholesterol esters, and triglycerides. Some enzymes are also concentrated in the Golgi fraction, like thiamine pyrophosphatase and glycosyl transferase. Cisternae, also called as lamellae, are most constant elements of Golgi complex, which are flattened parallel sacs situated upon one above the other to form stacks. Number of cisternae in a stack, it varies from species to species. Number of cisternae in stack is about 4 to 5 in most plant cells and also animal cells. In case of Euglena, this number may go up to 20. The membrane of cisternae is roughly 70 angstrom in thickness and having cavity of about 150 angstrom wide. In some plant cells, Golgi complex is polarized and has two well-defined phases. One forming phase and second maturing phase. The forming phase is outer side and maturing phase is on the opposite side. A maturing phase or secreting phase where lamella buds up secretory vesicles. New lamellae are formed on the forming phase and maturing lamellae are lost on the maturing phase. Cisternae lie parallel to each other and are separated from each other by a space of about 200 to 300 angstrom. Small vesicles are having diameter of about 400 to 800 angstrom. These vesicles are intimately associated with cisternae and may show continuity with them. Small vesicles arise from cisternae by budding. Large vacuoles are clear and they lie generally at the edge of Golgi complex. Now, what are the functions of Golgi complex? So it plays main role in the formation of secretion granules. It has also a role in protein secretion. Golgi complex acts as condensation for proteins formed on the ribosomes. Concentrated material is then packed for secretion. Golgi also plays a role in synthesis of carbohydrates. Enzymes and acid have been demonstrated to be active in Golgi complex. During maturation of sperm, Golgi plays a role in the formation of acrosome. In many mammalian tumor and cancer cells, Golgi complex have been described as a site of origin of pigment granules, for example, melanin. So this is overall survey of one of the cell organelle, that is Golgi complex or dictyosome.